So hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Joe Ferraiolo from Repligen.
Good morning, everybody. Thank you for joining us this morning. Thank you for coordinating this. This morning, we're going to be talking about using slope spectroscopy for faster, repeatable, more accurate protein concentration measurements. We'll be talking about the current challenges with UV spectroscopy, or traditionally called traditional UV, how the VP technology works, and then also discussing and presenting some information about customer-provided data and publications to support the technology going forward. As most of you know, Repligen Corporation is really involved with high-impact technology and innovation since 1985, most in the processing areas, production and pharma, and now with the introduction of C technologies, introducing the analytics portion to the catalog of items, really give the corporation a large view of how we can help and innovate this technology and processing moving forward. So what we have here is the first and only at-line processing analytics using slope spectroscopy. So to be very clear, it is a UV-based technology. We're not doing anything so radically different here. It is based in UV. It's just an updated and modern approach to measuring proteins and essentially OD readings using the VP technology. The results, obviously no estimations and dilutions or manual calculations built in. So the primary benefit of the Solo VP is no dilutions. However, having a slope-based technique allows us to provide more accuracy, repeatability, and easier implementation into how the technology is used. It's also adaptable to inline analytics. We are now included in the ASCM Standard as well. And to date, there's well over 1,000 systems installed throughout pharma on a global level. So the real approach to slope spectroscopy is trying to refine the process through an updated, sort of modern approach where we can get away from the essentially 7-step traditional UV process, which is not only time and labor intensive, but obviously has the error-prone dilution aspect involved, and really scale it down to essentially a measure report function in a matter of minutes. Most of you that are involved with traditional Beer-Lambert's law have obviously seen this in your history, where to make the measurement using traditional UV, or really any fixed path length UV, whether it be a plate reader or a nanodrop, it's still all fixed path length based. So you have to dilute down the sample in order to fit that sample within the linear range of your spectrophotometer. So before we've even tested any of the samples, we're introducing potential error from the very start. Reason being, obviously, limited detection range, linear range of your spectrophotometer, and the fixed path length that's available to you really only provide a limited window for linearity based on that sample. So if your sample happens to fit within the path length range of the instrument, then in most chances it might work for you. But as we know, in most cases, especially with high concentration samples, it does not. The obvious issues with this, assay time to prep samples and create a standard curve and back calculation can take anywhere from 30 minutes to 3 hours. So it's certainly labor intensive. You also lose out on time the more downstream you go in the process when the sample requires a transfer internally to other labs for GMP measurements to be made, and then the previous lab waiting on those results before they can continue the processing. All the time and dilutions associated with this are obviously a restraint on throughput and the accuracy of the measurement. And the dilution error can range anywhere from 5 to 20%, depending on if you were making dilutions volumetrically or gravimetrically. So really we want to try to introduce a technology that takes the two biggest areas of concern, which is dilution error and time, out of the equation, and really sort of address that challenge in a more robust technology. As I mentioned previously, the standard UV technique has many areas where the step processes are time limiting, and certainly with dilution error from scientist to scientist can vary. What we have done now is either removed or automated this entire process down to a measure report. So there's obviously no estimates and diluting involved with the solo VPE. For most MABs, the range of concentrations that the solo VPE can measure is from 0.01 mg per mil up to 300 mg per mil without the need of any dilution. Additionally, later in the process where you're plotting and calculating to acceptance criteria, all of that is automated within the software. So that's where the 1 to 2 minutes come in. And that includes the sample analysis, collection, and any acceptance criteria you're measuring against into the report. So we've now taken potentially half-hour to hour processes and reduced it down to essentially 2 minutes. Slope spectroscopy, again, still a UV-based technique. The difference in what we're doing here is we're varying the path length. So L is the variable in the equation, while the concentration is the constant, which is untouched. So rather than scanning across a sample compartment in traditional UV, we bring light through a fiber optic piece of material called the fibrette. So the light path is determined by the bottom of the fibrette and the bottom of the sample vessel. The fibrette is not hollow, it's not capillary. The liquid volume in the sample goes nowhere. This is purely a way from us to bring light from the spectrophotometer into the sample. The second thing we do differently is we take multiple path length reads in the measurement sample to calculate a slope regression. So the solo VPE is not an absolute absorbance-based technology. We actually don't care about absolute absorbance, meaning that things like fingerprints and scratches on the vessels have no influence on the sample measurement. The solo VPE only cares about the change of absorbance from path length to path length to generate an accurate slope regression. The same would apply for things like baseline correction as well, where typically most buffers and blanks have no absorbance change over path length, therefore can be removed from the experiment altogether. The way the system works, we provide an Agilent Carry 60 as our light source, and via a fiber optic delivery fiber, we bring the light from the carry to the solo VPE. No measurements are made within the carry. They are made directly within the solo VPE itself. Once you press the start button in the software, that piece of material called the fibrette will lower itself into the sample volume and set 0 path length. We equate this to carrying a scale to assure us that the fibrette is starting at 0. The software is then completely automated to scan the wavelength of interest from small path lengths to large path lengths. The software is actually looking for one absorbance based on the path length that's chosen. This process is completely automated. The user will not have to define path length range and path length step. The software is intuitive enough to know that if it detects a very high change of absorbance, it will measure very small path lengths. Other way around, if it detects very little change of absorbance, it knows it's got something very dilute and will auto-adjust to a very long path length step to generate that change of absorbance. Once it finds the one absorbance target range that we're looking for, the Solo will then plot between 5 to 10 path lengths worth of spectral data to plot a slope regression for us. Data acquisition takes about 1 minute. And the result is a slope-based concentration result. So now our equation is slope divided by extinction coefficient equals concentration. The path length range of the Solo is from 5 microns to 15 millimeters, essentially giving us 3,000 choices of path lengths to establish true linearity within Beer-Lambert's law. The R-squared value is our proof with every measurement that we have accurate and linear data. We have now taken flow spectroscopy and resolved it into a pass-fail test, provided your R-squared value meets our criteria of 3 nines or higher. So when we're plotting linear data, this graph shows that the Solo VPE will only use the most linear data it's able to generate based on its search algorithm, which is this green section here. It will never use potential absorbance data that's outside the linearity of the system that within spectroscopy we would never consider using within an absorbance-based measurement. And obviously the blue area represents saturation where dilution needs to be required. Faster, easier, built-in data quality ensures that the 3 nines of linear data gives us our accurate result. And because it's slope-based, we don't rely on user-to-user differences in concentration or repeatability, which is about plus or minus 2% on the system. A few years ago, we collaborated with Bristol Myers in New Brunswick, New Jersey, and how they validated the Solo VPE for in-process and clinical manufacturing on the manufacturing floor. The publication details the complete method validation for how they completed implementing Solo VPE within manufacturing. The obvious is that it's certainly comparable at the bare minimum to traditional UV. However, the differences they used to see with their gravimetric dilution at about 3.5%, they now see in less than 0.5% using Solo VPE between 2 analysts and 2 systems that happen to be in different manufacturing suites. They were able to refine the process from 3 hours down to 30 minutes. Additionally, the number of days saved is per Solo VPE. So we've never had a customer not save at least a month of time when implementing slope-based technologies over traditional UV, which then account for a very quick return on investment in approximately about 3 to 4 months' time. What's not in this paper is that BMS was able to fit an additional lot in at their first year of implementation and potentially revisit the number of shifts required on site now that the measurement's being made directly on the floor. Last on this paper, because the data was so thorough, a lot of our customers currently use this paper as their template when performing method validation within their organizations. This risk assessment was also published by Bioprocess International as well. It was a collaboration with the analytical and QC groups in Research Triangle Park, and truly understanding how much time and money they're saving using Solo VPE over traditional UV. And what they found was a cost savings of over 50%, which equates to about $300,000 a system in the article. So again, what is not captured here is any potential investigation in laboratory time. So because we're not relying on dilutions and sort of the human element here, investigational times are reduced and sort of pinpoint accurate to if there was a discrepancy in the results. It's much easier to know where that came from rather than having the traditional UV variables involved in the current process. This company saves nearly 2 months of time implementing solar VPE over traditional UV. Last on the paper side, we are now included in the ASCM Standard for general practice of UV techniques, allowing for the adjustment of path lengths when making any UV analysis. So not only have our customers across the board start to adopt solar VPE technology, that regulatory standards is now seeing this as a more robust way to do UV analysis. Currently, as we mentioned earlier, there's well over 1,000 solars used for various applications, even outside of A280 and protein measurements, such as DNA, polysorbate analysis, Planova filter testing, virus filtration, and we now have 35 commercial drugs and substances validated using slope-based spectroscopy. So the generated data is going to regulatory based off slope. Another 40 validated for in-process testing. 7 additional commercial drugs are in validation stages, and we've expanded into biosimilars as well using Solo. The next generation of VPE technology is our Flow VPE device for inline workflow applications. It works, operates exactly the same as a Solo VPE. The differences being that it's completely stainless and has a flow cell attachment for a variety of different sizes. It can be attached to the skid itself or put on a mobile cart that can be wheeled up next to the process for analysis. Chromatography, UFDF, fill finish, and mixing applications are the primary 4 that will be used for this technology. Users will now be able to make real-time decisions during the actual time of processing. So gone are the days of having to stop the process, pull sample, dilute down, see where you are within the process. You can now make real-time decisions using this technology. So again, a real change in how UV has been done. It's an evolution that's long overdue in modernizing this approach for protein quantitation. The idea is that this could be push-button analytics for even the untrained scientist or lab tech, that they will get the same result as anyone else that's properly trained on the instrument with even little to no knowledge of Beer-Lambert's law. It's certainly much easier to validate. It's being supported by ASTM now as well. And certainly the time and having a more robust method in place for all of your processing needs is certainly a more futuristic and, frankly, modern approach to how this is done. I think we can all agree that if traditional dilution UV never existed and the 2 choices were given now, either here's a process that requires dilution, here's one that doesn't, I don't think anyone would choose the method that requires the dilution. Again, my name is Joe Ferraiolo. Thank you all for your time. I understand how busy everyone is, certainly in this market, so it's truly appreciated that you took the time to hear the webinar.
Okay, great. Thanks, Joe. So the first question is, has this technique been validated in GMP environments such as clinical or commercial manufacturing or QC?
Yes, that's actually our biggest area of growth. 2 out of every 3 systems sold now go directly into GMP environments exactly for those processes.
And are there any additional UV applications the Solo VPE can be used for?
Yeah, the outside of A280 for proteins, I would say DNA, RNA is certainly one of the biggest areas that's being used. Polysorbate testing, we have 2 current publications available. truly deviating from how it was currently done into a much more robust method. So certainly those are available on the website for viewing. And clone-over integrity testing for virus filtration. And then in the coming months, we will certainly be looking at gene therapy applications as well.
Okay. What do you think is holding back the adoption of flow VPE from solo VPE?
Sure. Much like the solo early on in its process, no one wants to be first to implement sort of a perceived new technology. So I think that's where we are currently right now. The real holdback for us certainly is we're trying to adjust to frankly, the size of what is tolerable to attach to a skid and where it is within the process. I can tell you the biggest leaps forward with using flow VPE. You'll see certain publications coming out within a few months from companies like Janssen, who will be the first to implement the flow VPE within their processes. But I think it's just a matter of now having to revalidate the flow VPE slope-based process over the traditional fixed path-length sensor that resides in the tank. That's probably the biggest area of sort of restraint and where the efforts are needed.
Okay. So we do have a number of questions, but in honoring the time that we set aside for this, I'm just going to ask one more out loud. If you want to go ahead and type in additional questions, we're passing all the questions to Joe, and he can follow up with you directly after the webcast. So the last question for now is, presumably you require the molar absorbance coefficient in order to determine protein concentration. How does the technique work upstream with impure samples?
Yeah, if you require concentration, then you have to know extinction coefficient. There's no way around it. What the solo VPE will provide to you, if you have the zero information, is the OD reading, which would be the slope. So we have a lot of our small molecule users that have never had extinction coefficients, but they have quite the history of a robust library of expected concentrations. So the solo is able to generate a slope-based extinction coefficient based off calculated concentration. So you can certainly sort of flip Beer's Lambert's law upside down and do it that way.
Okay.
Well, I do see there's sure.
Yeah, there's a couple more questions, and we'll pass them to you, and you can follow up with them after. So thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. Look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.